Hello, everyone. Welcome back to our first section of a new exam material set dealing with the cardiovascular system. So, this is lecture number 1 of 2. This one's concerning the blood. I like to break them down into two-part lectures, even though. So, of this section, this is the easier of the chapters. The two hardest things in here are going to be blood typing and coagulation cascade to really be the hardest part. I'll kind of inform you on some things I'm going to be indicating here and expecting here. So let's begin things, of course, with a pint of sweat saves a gallon of blood. So the work you're putting in, the efforts you put in early will save lives. This is the goal. You don't take this. You don't study just to pass the test. You study because one day you're going to be the only thing between a patient and a grave, and that's what I'm here to prepare you for. So let's get into this. Let's look at it. Let's remind ourselves first off, chapter objectives. These things are always going to be tested on. So that means there will be a question on each one of these objectives on this exam. And every exam that I give, I will test on every chapter objective. These are the objectives the college set forward for me. These are not Professor Lefford's objectives. So let's take a look and see what's going on. And let's see what we can help you guys understand. So starting off, and I've got a few big things here. We want to start off with what blood is. Now, blood is a fluid. It's a connective tissue, which means this is going to be cells suspended in some sort of matrix. Remember, if you go back and remind yourselves that matrix is a component of usually protein fibers and a ground substance, and some ground substances are very fluid. Some ground substances are gelatinous, but depending upon the densities of that, so this is very fluidy, but very little fibers present, but a bunch of cells. Now, given that, this fluid connective tissue has 5 major functions to it. One of the first things we're going to see is it transports gases. What do we mean by that? We mean that this tissue, blood, carries oxygen and carbon dioxide between the lungs and the tissues. It carries gases to and from the lungs, okay? Then we're going to see it also carries nutrients, for example, from the digestive system, from your adipose tissues, your liver tissues, things like that, to the body's tissues. So it's going to be moving things from the digestive tract and fat, moving it to the tissues. And then it's also carrying hormones. It'll be carrying hormones. We saw in the endocrine system that hormones are chemical messengers released from an endocrine gland that secretes into the extracellular space, extracellular fluids, that diffuses into your blood and then is carried by the blood to the target cells. So that is a thing. It is a transport medium for gases, nutrients, hormones, and waste products. Now, the blood is the place where you add or remove ions and add remove acids from the ECF. It is the place, the primary location where we're taking electrolytes, acids and bases, and adding and removing them. Doing that at the kidneys, for example. Now, it also is a place where we restrict fluid loss. So, for example, coagulation of plasma, clotting of blood, things like that happen. So blood clotting and coagulation are not the same thing. That coagulation can form a platelet plug, help reinforce a platelet plug, but it is something different. Now we'll get into the slight distinctions between that. Now, it also defends against your pathogens. Now, how does it do that? They're white blood cells. White blood cells are going to be fighting your infectious materials, fighting viruses, fighting bacterial infections, fighting fungal infections, things like that, protozoan infections in the blood. And so they're going to be able to go in and make things like antibodies or other methods of fighting. We'll see that in the lymphatic system, how they fight, but they're going to fight infections. Then another thing it does is it controls body temperature, stabilizes body temperature. For example, if you walk out into the cold, you're going to see that blood may actually be distributed to that tissue on a short term, and then maybe as it gets really cold, it pulls it away from that tissue to keep the blood in the body, your core body temperature, warm. So what it does is it takes any heat, produce some skeletal muscle contractions, and sends that throughout the body, using it to warm the body, for example. So you can shunt the blood away from areas or send the blood towards areas to control the body heat. So it can stabilize body temperature by transporting the heat from muscle contractions produced as a byproduct of muscle contractions, sending it to other parts of the body. So that's the 5 big functions. Now, if I were to take blood, and what we would see if we take blood from the body, what you would do is you would stick a needle in a patient's vein, one of their veins we're going to learn about in the lab. We'll talk a little bit about them. And you're going to stick them, and then you're going to take that whole blood that would be in there. You're going to spin that down in a centrifuge, and you are going to get a sample of blood that is mostly going to be liquid called plasma, and then you're going to get red blood cells, RBCs here, and just a little bit here of something referred to as a buffy coat. And that's going to be WBCs, white blood cells, and platelets is your buffy coat. So we're going to see what that all is. Now, what we're going to do is give some relative abundances. First thing we're going to see, plasma is about 55% generally of all components of blood is plasma. So it's over half, so it's 55%. That is a value that we need to know. So it's 55%. So it ranges usually between 46 to 63%, depending upon how hydrated you are. So it's the liquid part, made mostly of water. It's yellow in color. It's got all the proteins, the cells, the ions in it, has a yellow coloration to it. Now, also, we said there are proteins in there. Proteins, these are going to be hydrophilic. What does it mean? Water, hydrophilic, loving. It's water-loving or water-soluble proteins. So most of these proteins are going to be globular proteins. They're globular proteins tend to be hydrophilic proteins, at least on the outside, hydrophobic on the inside. Carrying hydrophobic substances. So typically they have the hydrophobic exterior, hydrophobic interior, hydrophilic exterior. So these guys are water soluble, at least on the outside, their hydrophilic component. Inside maybe hydrophobic though. Now water is by far the most abundant. Now if you were to take the water here, and what we would do, would take plasma anyway, and we would take the clotting factors and deactivate them, remove them, go through. We have produced now blood that is incapable of forming coagulating plasma, because actually coagulation is a process of plasma. It will then it can no longer coagulate, has lost the ability to coagulate. We now call this serum. Now, other things that are in there, like your ions, like sodium ions, chloride ions, potassium ions, calcium ions, magnesium ions, phosphate ions, yada yada ions in the blood. I've just given a few examples. There's going to be ATP. Remember, ATP, adenosine triphosphate, will be a major component to that, as well as some other important things here like waste products. And the waste products, you're going to find things like urea, for example. You may have heard of blood urea, nitrogen, or BUN. Then what we're going to see is the formed elements. Now, the formed elements, the blood, this is going to be, guys, about 45%. So what you're going to see is this part down here, including buffy coat and the RBCs here. I was supposed to make that black. Is that 45%, and all of this collectively is referred to as formed elements, which all formed elements, red blood cells, white blood cells, and platelets, constitute 45% roughly. Now, it can be a little higher or a little lower. It can be about 37% to 54%, depending on how hydrated you are. The more hydrated, the lower the formed element percentage. The more dehydrated, the higher the formed element percentage. Okay? Now, we will see those formed elements are erythrocytes, leukocytes, and thrombocytes. Erythrocytes, red blood cells; leukocytes, white blood cells; thrombocytes, platelets. So we saw those are formed elements. That includes the buffy coat and the red blood cell layer. So the buffy coat's what's right in between. You would see in a spun down sample is a buffy coat. I always like to talk about the buffy coat. You will see it when you do when you learn about labs. Sorry, I had to take a drink. So we're going to see white blood cells fight infections. Erythrocytes that carries the oxygen, carbon dioxide, and the thrombocytes are involved in blood clots. Now, leukocytes have 5 different types, and an easy way to remember the 5 different leukocytes is never let monkeys eat bananas. Never let monkeys eat bananas, okay? Easy way to remember them. We're going to talk about them more in depth and later on. And here on the right side, I have a good image showing the breakdown of a whole blood composition. So what we're going to do is look at this. Now, how do we get this blood that I've drawn right here? So I drew this and spun it down, and this is your typical blood sample. And with that blood sample that I formed, how do we get it? Well, there are 2 ways we primarily get blood. The 2 major blood collection methods that you're going to be seeing is referred to as either a venipuncture or an arterial stick or arterial puncture. Now, venipuncture is the more common. This is the one that you're going to see a lot where we go in to a vein, usually right here, median cubital vein is the most common of the superficial veins that goes from your medial basilic. Okay, sorry, I got interrupted there for a minute. Okay, so you have your basilic vein, and which is the more medial, and then your cephalic vein more lateral, and bridging between those is median cubital. That's the primary place we're going to take it from. They're easy to locate. It's superficial, for example. Veins usually have a thinner wall, we're going to see. They're much lower pressured. They usually heal easier because of lower pressure. And what we are using them for is we are doing what's a metabolic panel, like hormone panels. We're going to look at how much your hormone production, hormone values. We're going to look at your insulin. We're going to look at your glucose levels. We're going to look at your certain levels of certain materials indicating your general overall internal health, things like blood urea, nitrogen, stuff like that. And so this is usually much lower oxygen content and much higher carbon dioxide CO2 content in this case. So it's also much lower in nutrient values because this is venous blood and it is typically not carrying fresh nutrients oftentimes. Now this can, depending, especially in a vein like this, if you were to look at a vein, say if you were to look at the superior and inferior mesenteric veins, that would be a different thing. About a portal vein, that'd be a very different content of nutrient. Now, next is arterial puncture and arterial stick. And what arterial puncture, arterial stick, this is sticking an artery. So usually we go to the radial artery, or for example, radial artery is going to be on the side of the radius, which is going to be the more lateral artery of the forearm. We might go to the brachial artery at the elbow. That should say brachial artery at elbow there. That's what that really should say is at elbow, but I must have missed that when I was typing in the notes there. So that's kind of my fault, isn't it? So that's usually things that I miss. And while I'm doing that, let me just go ahead and fix a few things. There we go. So I always hate that I did that, but it's just bugging me. And I'm like, oh, okay. I've always got that kind of bug. So what we're going to see, it is very high nutrient, high oxygen, low CO2. So because of the high oxygen, this is what we're doing with it. We are measuring, guys, the efficiency of gas exchange that is going on at your lungs. So this is when you hear about arterial blood gases, you collect arterial blood gases through arterial stick or arterial puncture. You may not know this, but if your respiratory therapy is what you're wanting to do with yourself, yourself, you do these. Now, when we collect blood, either through venipuncture or arterial stick, when you collect blood, blood's major things are basically this. There's a range and average to some things, but blood is slightly higher temperature than normal. Blood is for in America, if you're talking about the Fahrenheit system, 100.4 degrees Fahrenheit, sorry, or 38 degrees Celsius. 37.5 roughly is your normal Celsius for a human, 98.6 degrees Fahrenheit. So this is slightly higher than normal. Blood is 5 times thicker or more viscous. Think about viscous. If it's viscous, think about mucus. Think about snot. Snot is incredibly viscous material, so very thick. And then pH, blood ranges. If you have me for MP1, then you know that blood ranges from 7.35 to 7.45 in pH. That is our blood's range. It averages about 7.4 pH, 7.35, 7.45. I would say pay attention to that value. Now, let's just say a good average blood volume is about 5. For males, it's 5 or 6 liters, and females, it's 4 to 5 liters. And the difference in the 1 liter here is body size, body mass. Typically, males are bigger than females on average, but not always. You know, but on average, so we'll see it's about 5 to 6 liters in fluid volume for male, to about 4 to 5 in female. So you're talking about 5 liters would be a dead center kind of number there, average, okay? Now, plasma, this fluid, remember, this is the matrix of blood. It is, again, about 46 to 63% of the total volume of blood, which means it is about 55% average. Now, given that yellow color, it's going to be yellow because we're going to see there's a molecule in it called bilirubin. Bilirubin is going to be given its yellow color. That's what builds up in the jaundice patients. Jaundice being a condition that we'll talk a little bit about jaundice in a moment. Now, plasma and interstitial fluid, the fluid around capillaries. So if we were to get in, and let's say right here I want to explain it a little bit. We'll do this later on as well. Chapter 21 will be a place we'll talk about this. And but let's say I have a capillary right here. This was a capillary in the body. And right here you have some body tissues. Let's say this is some body tissues in the body. This might be some muscle tissues. Okay, we have tissues here. The fluid located in here between these is called interstitial fluid. Interstitium. Now, this fluid here, along with plasma, make up the major components of one's extracellular fluid. Okay, that's a lot of the extracellular fluid there. That's the major components, 2 major fluid compartments, we might call it. Now, there are 3 groups of plasma proteins that are suspended, and about 7% of plasma is protein. And of those 7%, so if you were to take the plasma, which is about 55% of the volume of blood, roughly, is plasma, take that out, all the plasma, and 7% of that volume of plasma would be protein. And of that 7%, 60% of that protein is albumin. Albumin is 60% of all the plasma proteins. It is by far the most abundant protein in plasma that you're going to have. And it's there to transport things like, when we talk about how it transports thyroid hormones, it transports some steroids, because remember, steroids are hydrophobic and they can't travel through the blood. So what they have to do is hitch a ride on a protein. And that protein here, guys, is going to bind to it and carry it through. So what you have is this transporting things like fatty acids, thyroid hormones, steroids, who are all hydrophobic. Now the next one is another set of globular proteins. We call them globins. Now there's many globins out there, transport globins, things like that. But they are about 35% of the plasma proteins. And they fall in two major categories. There's what we call the antibodies, which are a type of globulin. They are immunoglobulins, or Igs, to fight infections. Immunoglobulins, Igs, antibodies, they help me fight infections. They are globular proteins found in plasma all the time. It's normal to have antibodies. Antibodies may go up or down. Like that's what we're doing is if you want to check, see if you had your vaccinations before you go to your programs, one of the things you could do is either show that you've had the vaccination or go in for an antibody titer. If your titer is up, your circulating antibodies for that is going to be sufficient, showing that obviously you have enough antibodies to fight that disease. Now, transport globulins, they're going to be doing things like some of the any insoluble hormones, like transthyretin. We saw what major function was transport thyroid hormones. Transferrin transports ferrous, iron, Fe2+, ferrous, Fe2+, ferric is 3+. Apolipoprotein, which is what transports your triglycerides and other lipids, for example, cholesterol, is your apolipoproteins. You probably have heard of LDL, HDL, high density lipoprotein, or low density lipoprotein. So apolipoproteins, these are what transports fats in the body, triglycerides, things like that, because they are, again, water insoluble. Now, 4% of the proteins found in plasma, it's only 4%, but it's very important, is this word fibrinogen. Now, in your notes, I always say, anytime you see anything in Anatomy and Physiology 2, and the word and the suffix at the end is gen, underline it every single time you see gen at the end. Why? This is going to be indicating in your mind that if it says gen, it must be activated. Now, when I say fibrinogen, I'm saying it's inactive. You convert it to fibrin. Fibrin is what causes your blood clots. This is the strands that come in as blood coagulates to form part of your clot, to reinforce the clot anyway. Think of fibrin as the clot reinforcer, okay? I like to think sometimes as your platelets are kind of like bricks and the fibrin's kind of like the mortar, and you stick it in there to make sure the bricks stick and make that wall for a while. Now, that's only 4%. Now, you got about less than 1% is regulatory proteins. And these are going to be things like hormones, other proteins made by the liver that aren't there, antibodies, as some other antibodies as well, then other than just some of the antibodies that are already mentioned. There may be some other antibodies present, because you have two different classes of antibodies. There's antibodies from plasma cells, and then there's antibodies floating around the plasma that didn't come from those cells. Specifically, I'm talking about IgMs later on, things like that. So anyway, you got some other proteins. One might even say enzymes. There's a variety of things you can put here, honestly. Peptide hormones. Now, 90% of all proteins that are found in the plasma are made by the liver. If you really look at it, almost all of these come from the liver. So, fibrinogen, that comes from the liver. A lot of your transport globulins, they come from the liver. A lot of your globulins come from the liver. Albumin comes from the liver. And the liver being a source of this. So a lot of times what you will see is patients who have liver disease will oftentimes have blood diseases comes with it. Okay. Now, plasma also contains electrolytes. Electrolytes will be things that are dissolved. And this contributes to what we call the osmotic pressure. Remember, osmotic pressure means that if something is dissolved in it, that solution has high osmotic pressure because water wants to move towards it. So the more solute there is in something, the greater the osmotic pressure will be. So because osmosis, it's a real hoot. Water always moves towards a greater solute. So solutes, electrolytes, contribute to the osmotic pressure of body fluids, making water want to go towards it. Things like sodium, potassium, calcium, magnesium, chloride, HCO3 minus bicarbonate, HPO4, that's hydrogen phosphate, that's a type of phosphate ion there. Sulfate, SO4 2 minus sulfate ions. Some phosphate ions. So there's a variety of those dissolved in there. Now, other organic things you might find there. There's some organic nutrients found in there. There'll be fats, your fatty acids, your cholesterol, your glycerides, triglycerides, fatty acids, triglycerides, cholesterol, lipids, those type of lipids that we would find in our blood that are nutrient lipids. Then we would find carbohydrates, specifically mostly going to be glucose, and you'll find a slew of amino acid nutrients present. And there's a lot of other things in there. Now, also what you're going to see is some waste products. And this is where when something is broken down, these guys, you will have like biochemically, let's say I ate a hamburger and the enzymes start to break that down and biochemistry happens, you're going to start to see some protein, some things build up. You're going to have molecules like uric acid or creatine or things like that, ammonium ions, bilirubin, things like that accumulate into the bloodstream depending on what's going on. And we got to be able to get these places to where we break it down. Like we might take that urea to the liver. Liver might be able to take the urea and convert it to some things, uric acid, creatine, bilirubin, things like that, or take it to the urine and get rid of it. Pretty much, there's two ways to get rid of everything. You defecate it, you urinate it, or you sweat it. Pretty much, it's really it. Now, these things are either going to be taken to the liver to be broken down or excreted by the kidneys. So that's what the blood's carrying, these organic waste to get rid of it by breaking it down or excreting it to the kidneys. So it acts as a shuttle to get it there. Now, we said that about 45% of overall blood is red blood cells. Now, so it is 45% of the blood, and it is about 99.99% of all the formed elements. So the formed elements, this is huge composition. Now, though red blood cells are a little less than half of blood's average volume, about 45%, it could be more. It could be about 52% if you were dehydrated. That means, guys, but of the formed elements, normally a human should have about 99.99% roughly of their blood's formed elements will be red blood cells. White blood cells, never let monkeys eat bananas, fight infections. I don't have it in that list here. I think I had it in the list from here this time or something, but never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, never let monkeys eat eosinophils and basophils. Now, there's also platelets used for blood clotting. Now, platelets and white blood cells combine together, and they are only about 0.1% of formed elements. So right here, guys, when you look at that, of that formed elements, this guy here is only about that little bit, that 0.1%, 0.1%. Okay? It is a tiny little fraction of what overall is. So 45% being formed elements, and then here red blood cells, remember they are about 99.99% of overall formed element. So that's quite a big difference, okay? Now, all right, so hopefully we're good on that. So what we're going to do is continue on talking about now red blood cells being 99.99% of overall thing. How do we get them? Well, let's kind of look at this like it was a story. Now, in a story, every hero has a beginning. Our hero here is red blood cell. So red blood cell is kind of a hero. And red blood cell, he starts off in red bone marrow and like the end of a long bone. Now, what's going to happen is this little guy, when he is first born, a little red blood cell is born. What happens is there is that red bone marrow, and there's a cell inside of here, and this little cell, let's label him down here, is called a hemo blood cyto cell blast maker. So what does that mean? It means a red blood cell maker, okay, or blood cell maker. Let's just say blood cell maker. That's a little bit easier. So this means he's a blood cell maker, hemocytoblast. And this little guy will go around and live about 120 days. Okay, that he's able to live. So what happens is the cell goes in, the process of forming it. Now this begins at birth. Now in the adult, cells are going to be going on in your bone, but your first blood cells are being made because you need blood in the fetal bloodstream about third week of development. Now third week of development, you don't have bones. Because you need blood to make bones, don't you? When we learned about, we learned that in AP1, we saw that, we saw how endochondral ossification happened, and we saw how you really can't ossify bone until blood gets to it. So if you need blood to make bone, but you need bone to make blood, well, here's the problem. How do I do that? Well, in the bloodstream, you have embryonic blood cells. You actually got those from Mama. Mama gave you those. And these cells, these embryonic cells, they go to the liver, the spleen, the thymus, and the bone marrow. And those cells become stem cells that can make blood cells, okay? Make blood cells. And they are referred to as hemocytoblasts. Now, right now, the books are going to say the only site for red blood cell production in humans after birth will be the bone, the red bone marrow. There is some evidence to suggest otherwise, but for now, there's some evidence to suggest we might also make some in lungs. Now, there is some white blood cells that are also made there. White blood cells, many of them are made there. We'll get a picture by the time we get to the lymphatic system. You'll see all of it, okay? So what we want to do is we have a cell here, and kind of think about this like it was a family tree. Now, you have a hemocytoblast. Now, sometimes I like to think about hemocytoblast, and hemocytoblast can have some kids. Now, there's one or two kids he have. He can have a myeloid stem cell or a lymphoid stem cell. But it's an either-or situation. A hemocytoblast can make a myeloid stem cell or a lymphoid stem cell, okay? Myeloid stem cells will go on to make red blood cells, RBCs, and WBCs. Let's write a caveat, not lymphocytes. I can't write with my iPad sometimes. Not lymphocytes. Lymphoid stem cells will make lymphocytes. So now we've got red blood cells, white blood cells that aren't lymphocytes, and lymphocytes. That is all formed elements almost, isn't it? Let's see. RBCs, most WBCs, no lymphocytes. Now, lymphoid stem cells will go on to make lymphocytes. Now, I need to talk a little bit about that here. There's a diagram I have, I think, right here, down. Maybe I don't have any more. Here, myeloid, and what I really wish I had done is also mentioned platelets here, because it does do that, and that was something I wish I mentioned here. To be honest with you, let me, and what I'm going to kind of do, just because if I don't, I'll forget it, and say, and platelets. And if I do that now, it'll be there. So that's my fault. I'm just going to kind of add in there. I will not test you on it on this if I ever ask that. It's my fault for leaving it out, honestly. Now, lymphoid stem cells, they make lymphocytes. All right. Now, erythrocytes, what are we going to see happens to these guys? So remember, we got a hemocytoblast, and he's just there waiting and willing to become a red blood cell. Now, what's going to happen is he is going to need to be turned into, we need to make red blood cells under very specific conditions. Now, one of those things is if we have a low red blood cell count, anemia, or low oxygen count, which is referred to, guys, as hypoxia. Then the chemoreceptors in the kidney can detect this O2 deficit, and that will cause the kidney, who is monitoring blood levels of oxygen, will release a hormone called erythropoietin. Remember, EPO stands for erythropoietin, or it stands for what is called erythropoiesis stimulating hormone. And that's what we're studying. Is erythrocyte production is called erythropoiesis. Now, once this happens and the red blood cell production goes up, you will get more O2 because why? Well, how am we going to get more O2? Red blood cells go up. More red blood cells, more oxygen. More oxygen, O2 saturation, O2 sats go up. And when that happens and the O2 levels are going up, the chemoreceptors are going to say no more EPO, and EPO stops erythropoietin. Now, EPO, you may have heard of Lance Armstrong when he was winning the Tour de France, 50 billion times in a row. He had gotten in trouble for doing something called blood doping. And what he was doing was taking EPO, erythropoietin, and shooting it into himself, and that was able to get his red blood cells to go up, which meant he had more oxygen carrying capacity, which meant his tissues could get more oxygen, can make more ATP, could be able to do more muscle contractions. Now, red blood cells do something very interesting. Juvenile cells, when they're juvenile, they have a nucleus. And what the mature cells will do is they will anucleate. They yeet out their nucleus. They just kick their nucleus out. I oftentimes like to think about that is that red blood cells are somewhat like, I always think, why do mobsters like to drive big Lincoln Town cars? Because there's plenty of room in the trunk. So what they see is, guys, is the red blood cells here. If you get rid of your nucleus, it's like removing the spare tire out of the back of the truck in your trunk, and you've now got more room to put groceries in there. Okay, you got more room. So that's what we've done here, is mature red blood cells anucleate, kick out their nucleus. They lack a nucleus, and they also lack mitochondria and ribosomes. So they can't divide. They can't make proteins. They can't repair themselves. They can do very little. Now, you're like, how do they get ATP if they live 120 days? Well, they get it from the fluids they're around. The blood carries ATP. Everything they need, they're getting it from their own blood that they're in. Okay? So they don't divide, and like I said, they're going to live about 120 days. So we're going to talk about that here. So now what happens is 120 days is about how long this cell lasts. Red blood cell is made. He lives for about 120 days, Now, 120 days later, he is old. He's old. He's aged. He's damaged. He's floated around your blood vessels. He's smacked into artery walls. He's been damaged and hurt. And he's just old and worn out. So what do? We're going to send him to the liver, spleen, and bone marrow. And there are macrophages who will phagocytize him. So we bring him on into the liver, the spleen, the bone marrow, and we bring him in. And what we're going to do is we're going to take this little red blood cell, guys, and we're going to phagocytize him and break him out. And we're going to break down something that's called hemoglobin. Now, hemoglobin, so if you were to look inside of a little red blood cell right here, here's a little red blood cell, a little disky little red blood cell. And if I were to look inside of a little red blood cell here, what I'm going to do, if I were to zoom in, I would see a bunch of proteins here. And I would see hemoglobin. Now, hemoglobin, sorry, about to put the wrong letter there, is made up of two things. There is a globin. So you're like, well, where's the heme? The heme binds to the globin. So it kind of is like this. Imagine, I'm going to try and draw it this way. If we were looking at it from the side, you would have this large protein complex, and then sticking up out of it would be the heme, kind of a little disky-like structure. I always like to draw this little disc. Heme has this very complex kind of ring structure to it, is the heme, and it's sticking out here. And we'll talk a little bit about him. But the heme and the globin here, what this does is your heme is the thing here that your O2 will bind to and carry your O2 molecule on that heme. And I'm going to talk a little chemically how that happens. But what we're going to see is the heme is the pigment part. This is the part that makes blood red. Globin is going to be the protein part. Now, if we were to take a heme here and we were to look at this heme, and I said I'm going to kind of discuss it like it's a disc. If we were to look in this disc, at the center of this disc, we're going to see Fe++, Fe2+. It's at the center. So all the rest of it's kind of holding that together to the other parts of this molecule. And the iron is in the middle. Now, it's this iron here that is able to bind to oxygen, O2, okay? So your heme has an iron in the center here called Fe2+. We can say Fe2+ or Fe++ is found there, and the O2 binds to that, okay? Now, this O2 here binds to this, the iron. So what we got to do now is the iron is in the heme. We want to break all that off, and we're going to release the iron. So if we were to come in and take this heme disc and take the Fe++, we need to grab it up with a protein. Let's use purple. And there's a protein that will grab that up because it could go somewhere else called transferrin. And transferrin is going to go on to things like your liver, for example. So what it's going to do is transferrin is going to take this thing to your liver where it can recycle that iron. And that iron goes to transferrins, the different transferrins. And it can take that also, another place it might go is red bone marrow, and it can take it to red bone marrow where you make more hemoglobins for more red blood cells. So it'll grab it up. It might go to the liver. Some of it some of that iron from the heme gets reused, stored in the liver. Some of it also ends up in the transferrin, ends up in the liver, into the bone marrow, and that's added to it. Now, the heme is left. Now, we have this heme left. We got the iron, and we dealt with the iron, sending it to like the liver. Now I've got this heme left. Now, heme, if you take heme, which is a biochemical, we are going to take this heme here and we are going to convert it to biliverdin. Biliverdin. Now, biliverdin will go on to be broken down into something. Let's use yellow to make bilirubin. Bilirubin. So biliverdin, the green pigment, goes on to be turned into bilirubin. The yellow pigment may be a little orangey, and in jaundice patients, this accumulates in their blood and in their tissues, and that makes their skin turn very yellow. Now, biliverdin and bilirubin go out in bile, goes out in urine, goes out in feces. Bile is green because it's got a lot of biliverdin. Urine is yellow because it has a lot of bilirubin. Feces is brown because you take yellow and green and mix it together. What do you get? You get brown. A lot of the color for feces is because of this. So if you ever were to see blood, if you ever to see urine and feces colors change dramatically, you're going to see that can indicate a blood disorder. Something's going on. Maybe, some kind of blood issue going on, some kind of blood cancer or something like that. So you're going to see that's an indicator when you see blood. That's why you might see babies have very yellow feces, for example, getting rid. A lot of babies are born with excess bilirubin. Bilirubin. That's actually because of a condition. I was born with it. It's because my AB blood type is incompatible with my mother's AB blood type, and I was jaundiced because my mom's antibodies crossed the placenta that I was attached to and attacked my blood cells. Wasn't enough to really kill me or anything, but it was enough to cause me to be jaundiced when I was born. So a lot of times jaundiced babies it's a blood incompatibility. Now there is a very severe one we're going to talk about in just a little bit. So as I said, I've made it. My red blood cells, my hemocytoblast, became a myeloid stem cell. Myeloid stem cell here goes on to give a red blood cell. We release it. It goes out. It lives about 120 days. It gets old. Now, one of the things that can happen, you could be right here. Blood cell going around, somebody punches you, that cell ruptures due to some reason, hemolysis. Maybe that happens because of an injury. Maybe it happens because of chemistry. Maybe you have hypotonic environment in that area. We can recycle it there and get rid of it. And when a lot of the color changes, like the greens and yellows that accumulate, that's what causes bruising, is when it happens in the fluids. But normally our red macrophages in the liver, spleen, and red bone marrow get it, and they take this guy, they break him down. They get the heme, they take the heme, and then they take the globin and recycle it, reuse it. They take the heme out, take the iron and recycle it, send it to make new blood cells. They break that down to biliverdin, bilirubin. Now, bilirubin typically goes out to feces. Biliverdin usually goes into bile. Things like that. And we're showing bilirubin, but there's also biliverdin. Biliverdin is oftentimes defecated as well. So bilirubin may go out. Bilirubin may bind in the bloodstream, things like that. So you get molecules that go out into the feces, giving the brown coloration, things like that. Okay, so red blood cells, and then what I'm going to do here is probably after I do hemoglobin, I'm going to stop after that. We'll do the last part. Now there's lots of things you need. You do need proteins to make blood, but one of the two big things you guys need to get in the diets to make good red blood cells is vitamin B12, so the blood cells can grow. This vitamin is needed for certain enzymes necessary for production of red blood cells, and iron is needed for the hemoglobin to bind to the oxygen, so you got to get iron. So both of these things cause anemia. Low iron, low B12 cause anemia. All right, now red blood cells, remember, have hemoglobin. Now, hemoglobin is the red thing that causes blood to get red. Blood's red color comes from this protein, hemoglobin. Now it carries oxygen, O2, it carries carbon dioxide, CO2. And just to let you know, if I were to take one drop of blood and drop it out of a needle, there's over 1 million red blood cells in there. And they all live 120 days, and they all have their red blood cells. Now, let's get in here and talk about something. So we've done a drawing already. Let's do it one more time. If I were to take whole blood in a tube and I were to go and shake it down where I were to say, okay, this 55% plasma here, and then you had your buffy coat, then you had your 45% RBCs here. This 45% RBCs that will be floating down here in the bottom, that is down here at the bottom in a spun down, centrifuged down blood sample, is referred to as a packed, because it's been centrifuged, cell volume. But another name we give to this, which is more commonly used, is hematocrit. Hematocrit. Now, let's just take two of these things here and let's see if we understand. Now, what is hematocrit? Hematocrit is the percentage of whole blood composed of red blood cells. So here I have the percentage of whole blood composed of red blood cells, which in men is around 42% to 54%. Typical male's hematocrit is 42 to 54. Okay, now that is the percentage of a centrifuge blood sample that is composed of red blood cells. Now, right here, so let's take this one here. Let's say I've got this individual here, and their RBCs are here, and this individual's RBCs are up here. Or let's just do this. Let's do these the same and do something a little bit different. Sorry, let's do this. Let's put these RBCs at the same place. Now this patient, I go in and I get some thing here, this patient here. Okay, so if we were to take this patient and we looked at this, this right here is overall more percentage than this is. This is less a percentage. So this person here has a lower hematocrit. This person here has a higher hematocrit. Hematocrit goes up with dehydration. Hematocrit goes down with overhydration. So what you would be looking at is that somebody with less blood volume has overall. So if you were to look at these tubes here, getting a little bit better, and this is a little bit more skinny like our hematocrit tube. If we took blood and this person's plasma was like these two were here, and their plasma was both the same volume, we just had the same volume of blood, but that we took out the patient, you would see this patient here has overall less plasma than this patient has more plasma, and this patient here had more red blood cells, so they have a higher and increased hematocrit. This patient here has decreased hematocrit. The percentage of whole blood composed of red blood cells. So if we took the same volume of blood out of a patient who was dehydrated, they would have a very high hematocrit. Okay, that's to see if you can understand it. I'm going to give you kind of a little bit more of a clinical scenario there. Now remember, there is that buffy coat in the middle. Now, males have more red blood cells, higher hematocrit. Females' hematocrit is about 38 to 46%. Testosterone causes red blood cell production as well, and also just overall size. So if you took the same vial, now there is a platelet white blood cells called a buffy coat that's there as well in your tubes. Now red blood cells, they are kind of like squished donuts. It's they don't have a hole in, but they're squished. They're thin in the middle, thick on the edges. Imagine a donut that doesn't have a hole that goes through it, just a squished donut. So it's thin in the middle, thick on the edges, increasing the surface area. That this has more surface area than just a flat disc. Now, there's no nucleus because the nucleus leaves when it's matures. There's no mitochondria, so you can't make ATP except through glycolysis, which was an anaerobic or without oxygen process. And these will stack up and form a Rolo. Think about Rolo is candy you might get, a bunch of little chocolates stacked together, and they go through narrow cavities, and then there is the inside, there's a volume. I can measure this volume, and the volume in here is called the mean corpuscular volume. The mean corpuscular volume is the volume of a red blood cell on average red blood cell volume. Some folks might have larger mean corpuscular volumes, larger red blood cells, or smaller. But red blood cells have, you can see some are bigger, some are smaller slightly. They're not all 100% perfectly the same size, but they're very close. Now, hemoglobin. We said that hemoglobin was here. We said you have your hemes look like little discs with an iron in the middle. And this is what I mean by the discs. It forms this very complex ring structure. It's just for your edification. If you ever had to know, it's called a porphyrin ring. These little guys here all come together, form a ring with 4 little bonds around here, and this Fe2 plus binds to the oxygen. And now the heme, it goes on the globin, and the globin, there are 2 globin chains, 2 types. There's what we call alpha chains and beta chains. There are 2 alpha chains, light purple, and they're the blue or purple, and the blues, beta chains. 2 alpha chains, 2 beta chains. You will see alpha, beta, in a ring. And it does that on purpose. There is a really good reason for that if you ever had to learn the biochemistry of hemoglobin. Now, these 4 chains go in together, and they have their heme to bind to the oxygen. Now, when it's bound to oxygen, when hemoglobin has oxygen on it, it's called oxyhemoglobin. When it releases oxygen, we can call it deoxyhemoglobin. If it binds to CO2, it's called carb for carbon dioxide, carb aminohemoglobin. Why is it called carb aminohemoglobin? The CO2 does not bind to the heme. It binds to the protein chains, the amino acids of the globins. That's why it's called carb aminohemoglobin. So we can see 4 chains. Now here, all 4 chains have a separate color: yellow, blue, mauve, reddish, green. The gray circular structures, the orange is the iron, around with the blues, those nitrogens. And the 2 little red balls here, that's oxygen, that's O2. So that's an O2 bound. This is oxyhemoglobin, 100% oxygen saturation here. And when you damage a red blood cell and hemoglobin comes out, it breaks down in the tissues, causing the yellow, blue colors, green colors, things like that, and bruising. So what we're going to do is we're going to stop here. I'm going to pick up with part 2. So go ahead and take a break, stop, catch your breath, come back to this for another half of the week, but when you prepare for those examinations and things like that. So guys, thank you so much for watching. If you do have questions, don't hesitate to contact me, please. Thank you very much, and I hope you're doing well.